শর্মিষ্ঠার ছবিটা ঘুরে আছে।
ঠিক আছে,
হ্যাঁ
ঠিক আছে। হ্যালো,
না ঘুরে আছে,
ঠিক আছে,
ঘুরে আছে।
আচ্ছা দেখছি।
ক্লকওয়াইজ 90 ডিগ্রি ঘুরবে।
স্টার্ট করছি।
হ্যাঁ হ্যাঁ
Good afternoon to you all.
ছবিটা ঘুরেই আছে তো।
এমনি এদিকে ঠিক দেখাচ্ছে, এদিকে
এখন বলে নাও।
এবার ঠিক আছে স্যার?
হ্যাঁ 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 ঠিক আছে,
এবার ঠিক আছে।
Good afternoon to you all. Respected OIC sir, Dr. Shobhan Yogi sir, President and Head Dr. Pintu Banerjee, IQAC Coordinator Dr. Amoresh Mondol, IQAC Member and Head of the Department of Mathematics Dr. Kalidas Das sir, and my other colleagues and my beloved students. I welcome you all to this webinar. Today we are going to organize the webinar entitled A Voyage to the Industrial and Environmental Microbiology. Last few months, since March 2020, we are facing a pandemic, COVID-19. The world is going through a massive economic loss and human death during this pandemic. The world is in search of salvation or redemption from this pandemic situation. The immediate answer is vaccine, which is an outcome of biotechnology or medical biotechnology or medical microbiology or broadly industrial microbiology. A large part of pharmaceutical industry is included in this industrial microbiology, which can save the human life as well as show the direction of economic growth. With the industrialization, urbanization, over increasing population, and economic growth, the condition of environment is deteriorating. To determine the environmental degradation and to combat the environmental deterioration through bioremediation, the knowledge of environmental microbiology is essential. So to keep healthy environment and healthy life, these two fields of microbiology, namely industrial microbiology and environmental microbiology, is very important in this COVID-19 pandemic situation. This webinar is very pertinent to this topic. So in broad sense, our objective is to inculcate the preliminary knowledge of these two subjects to our students and at the same time get acquainted with the syllabus of DAC as prescribed by UGC and the University of Kolkata. We will organize this industrial and environmental microbiology subjects in 4 lecture series. This is our first series. I hope the students will enjoy the lecture. Thank you. Now I would request Dr. Shobhan Yogi, our OIC sir, for an warm welcome address. Over to OIC sir.
Thank you, Dr. Maiti. Am I audible?
Yeah, you're audible.
Okay, thank you. Today is the 25th webinar of our college. The journey we started from the first week of July to 2020, and within a span of 2.5 months, we have been able to organize 25 this type of webinars where renowned professors, scientists, doctors from our country and abroad have talked for us, for our students, for the teachers and community as a whole. So as a continuation of that, today our botany department along with IQAC of the college is organizing one webinar, a voyage to the industrial and environmental microbiology, a very appropriate topic at present situation. Today our speaker, Dr. Sanjeev Pandey, is from BB College, Asansol, West Bengal. I heartily welcome you, Dr. Pandey, in this webinar. I also welcome our beloved students who have joined this webinar, our teachers from the college, and other participants from other colleges and institutes. Our college, which is going through its 175th year of establishment, since November last year, faced a lot of problem due to the lockdown situation with the academic activities along with all other colleges and universities in the country. But using the technology, we have been able to continue our academic delivery to the students and to the community and to other people who are available these facilities.
Thank
Now,
you, sir.
I once again welcome Dr. Sanjeev Pandey, and I now hand over the microphone to Dr. Maiti for continuation. Thank you.
Sir. Thank you very much. Now I request our IQAC coordinator, Dr. Amoresh Mondol, to say a few words on this. Over to Dr. Amoresh Mondol.
Thank you. Good afternoon. A respected officer in charge, our distinguished speaker, his colleague of the Department of Botany and all other departments of the college, my beloved students and interested participants of this webinar. It is my honor and privilege to have been asked to say a few words in this state level webinar on a voyage to the industrial and environmental microbiology organized by the Department of Botany and the Internal Quality Assurance Cell, Krishnagar Government College. On behalf of IQAC, Krishnagar Government College, I must express my hearty congratulations to Dr. Pintu Banerjee, Head of the Department of Botany, and his departmental colleagues, Dr. Shormishta Maiti, Dr. Rituparna Kundu Choudhury, and Srimati Somanjana Khatua for
আমি কি কানে নিয়ে দেখলাম স্পিকারটা তখন শোনা যায়।
Thank you, sir. Hello? Hello? Amoresh da, আমাকে শুনতে পাচ্ছেন? Amoresh da, can you hear me? Amoresh da, can you hear me? অমরেশ দাকে শোনা যাচ্ছে না। অমরেশ দাকে শুনতে পাচ্ছ? আচ্ছা,
অমরেশের ছবিটাও ফ্রিজ হয়ে গেছে।
তাহলে লিভ করে।
নেটের প্রবলেম, আমারও তাই ছিল, প্রচন্ড প্রবলেম হচ্ছে।
আচ্ছা, তাহলে আমি থ্যাংক ইউ স্যার for saying a few words. And now, before I go to the mainstream of this webinar, I must introduce the resource person, Dr. Shonjit Pandey. Dr. Shonjit Pandey, presently he is an associate professor in botany at Banwarilal Bhalotia College, abbreviated as BB College, Asansol, West Bengal. He has a teaching experience of almost 20 years and a research experience of 24 years. He's an MSc Gold medalist from Bardwan University. He has qualified CSIR GATE and West Bengal Slate in 1997. He has many research projects, one of which is a minor research project of UGC, which he has completed, and one major project of WBDST. It is in the final year. Now he has a research scholar, one registered for PhD and two yet to be registered. He has a number of books. He has published Advanced Botany, Volume 1, second edition, and Volume 2, now in press. He has research articles of 17 national and international publications. And his present area of research is leaf litter and soil microbiology. And over to Dr. Shonjit Pandey.
Thank you very much, Shormishta.
Thank you, sir.
I also thank Principal Sir and Dr. Pintu Banerjee for giving me this opportunity to share my work with you. So, I should go to the presentation, I think.
Yeah, you can go to it.
Can you hear me properly?
Yeah. Yes, sir.
Okay.
You can continue.
Well, though you said that the industrial and microbiology with respect to the COVID-19 situation, my presentation will be not directly linked with that, but you can indirectly link it.
Okay.
Are the slides visible?
Yeah, no. They're not visible now.
Not visible?
Yeah, now it's
Yeah. Okay, let me go to the full screen. So, as my present area of research is about leaf litter microflora, and it is about a sal forest situated about 45 kilometers from my place of living, that is Asansol, and my college also. And this is regarding the leaf litter that fall on the ground on the forest floor. and they gradually decompose into soil, and how the microorganisms are involved, and they're converting the leaf litter from day zero to the day it is converted into soil. The story is about that. And but since the listeners are many students, I should talk a little bit about the basic concepts of microbiology also. So starting with this lecture will be in two parts. First, a brief introduction about the subject, that is microbiology, as Dr. Maiti said, about the industrial as well as the environmental microbiology. There are various aspects of microbiology actually, and many people work in different areas. And today's focus of this talk is about industrial and environmental microbiology, and this work is also related with these two things, along with this agricultural microbiology also, you can say, up to some extent. So what is this industrial microbiology is about? Industrial microbiology deals about the fermentation technology. Whatever the fermented products that we consume, like the antibiotics, the beverages, the syrups, etc., they are the products of fermentation, the industrial microbiology. The preservation technology that is used for in the food industry and other different kinds of materials also involves some sort of industrial microbiology. If you talk about the environmental microbiology, there are various aspects. Among them, where microbiology is the fungi and the bacteria and the other microbes are involved, it includes areas like bioremediation, biofertilizer, biopesticides, soil, air, and water microbiology. This is a huge, it includes every atmosphere, everything in the environment. So I will be concentrating on these red marked areas, the bioremediation, PGPRs, and soil microbiology to be the most. So I concentrated on soil microbiology a few years back, thinking that the soil is very important in present context when it is shrinking day by day. Hugh Hammond Bennett, who is known as the father of soil conservation, he has quoted this sentence that out of the long list of nature's gifts to human being, none is perhaps so utterly essential to human life as soil. As you know, soil gives us plants, and plants are the source of life. So what is soil? How can you define soil? Chemically, soil can be defined as it's a complex mixture of minerals, water, air, organic matter that is present, and countless of microorganisms that are present in decaying organic matters or living matters and mineralizing them and mixing them in this environment. The soil comprises of these things. So soil, the earth crust is not exactly the soil. It is the skin of the earth crust, you can say, the top soil, which is cultivable. In other words, you can say the soil is a thing that is capable of supporting plant life and is vital to all life on the earth, because all life depends on plants, basically, and microbes, because microbes were the starting point where from life began. So this is a picture which shows the different layers of soil. The top soil is this horizon, this O horizon. Above that, there is the top soil. That is the leaf litter also is present there. A lot of the plant roots penetrate there. In fact, the plant root penetrates below the topsoil. It reaches up to where the parent rock is being converted into, is being withered, and it forms this soil. But it is nutrient-less unless until the nutrients that leaches into this material, withered material, it will not be able to support plant life or any other life. So, but this topsoil, it takes about thousands of years to form about 1 inch of the topsoil. And to make it fertile, it further takes about 3,000 years. So it is very difficult to get a topsoil of 1 inch, how much time it takes. But we have contaminated this topsoil in 100 and 200 years because of industrialization and various other things. And That's why it has become so important. Factors that have contaminated soil are like this: soil erosion, industrialization, grazing, agriculture, deforestation. You are all aware about these things. I'm not going to discuss that. So how can we can protect our soil? Instead of, I have written is SOS, save our soil. Instead of that, save our soil, I should quote like this. So prevent degradation, reduce erosion, and conserve soil. That should be the strategy if we want to survive, if we want to sustain our life on this globe. So what are leaf litter? How forest leaf litter is related with this soil, this thing? The forest floor is one of the most important sites where decay of organic matter is taking place, its mineralization is taking place, and a nutrient cycling is working there. The nutrients that are formed in the forest floor are transferred to distant places, areas, through different water channels present in the forest, the riverine system, and the streams. And they reach to the different areas, and they saturate, they make the soil fertile, the topsoil fertile, where we cultivate our plants. Therefore, management materials is very, very important, and it must be maintained in order to conserve continuous supply of compost-like fertile soil. So up to this was the introductory part. From here, I will enter into what we are doing regarding this leaf litter microflora, how we are proceeding in our research, and how far we have been successful to understand the process. So first of all, if you want to do a leaf litter microbiology, you need to have, if you want to do a realistic research, in situ research, you need to have a area, have a leaf litter, sufficient amount of leaf litter in certain area, and that can be found definitely in a forest. So I selected the forest, which is called Gangajal Ghati forest, which is present in the Bankura district of West Bengal, and it is nearby. This is the Bankura district, this is Bankura, and this is Asansol. So it is the forest is nearly here. You can say this is Durgapur. The forest is at this juncture. In this diagram, it has been shown here in this map, enlarged map. And when we travel from Asansol, we first reach Raniganj, and then we cross Damodar River, we reach Meja, and from Meja, when we go towards Bankura, the forest falls in between, about 45 kilometers from Asansol. So it is basically a small forest, but it is good for the study of this kind because it is also facing human interference a lot. So this is a satellite picture of the forest. The forest, this is the area of the forest. The Gangajal Ghati forest goes like this. I have demarcated it like this for better understanding. And it can be divided into several subsections like Has Pahari forest, Devali forest, Latiamani forest. And this is the thermal power, Mejia thermal power station that is present here. And this is the ash pond the Mejia thermal power generates. The ashes and which were once a big forest has been almost being shrinked to a very small area, maybe about lengthwise it will be about 8 kilometers, and widthwise it will be about 4 kilometers or so. So, and this is the man-made forest. What happens when industrialization takes place, the forests are being cut out. But the industrial people, they say that they're asked to compensate the loss of the plants. And for that reason, they afforest a particular area, equivalent land. But while doing so, they actually cultivate a single kind of plant. Either it is eucalyptus or acacia or equiloformis in West Bengal, and in fact in all over India. So that is nowhere equivalent to the existing pristine forest that has huge biodiversity. So I thought at the beginning, before going to the leaf litter microflora, I should be understanding the nature of the forest. What is the biodiversity? What kind of leaf litters are being present? Because the composition of the leaf litter will definitely affect the entire process of soil conversion or whatsoever. So we first did a plant community analysis of the entire forest. These dots are the areas. These are basically quadrates. There is a process in ecology, quadrate analysis, by which we understand the biodiversity of a particular area. So there are 40 quadrates where lengthwise they were considered a kind of a gradient study. From road, this is the road. On both sides of the road, we took about this side about 20, on the other side about 20 quadrates, and also take some, this is a man-made forest. This is eucalyptus plus half part is eucalyptus and half part is acacia. That is called sonajuri in Bengali. So this was kept for comparison. There is a region beside this ash pond that is, you can call it the semi-man-made forest, because some of the plants that were planted by the industry, because of the thermal power authorities, they were of mixed nature. They were not simply the eucalyptus and acacia. There were some Delonix regia, some neem, some bale, etc. There were about 8 to 10 different kinds of plants, along with the forest plants. They were planted in the lines of this ash forest. That I'm calling as semi-man-made forest. So there was a comparison done between this pristine natural forest and semi-man-made forest and the man-made forest. I'm not going to that detail because it will take too much time about that. I have to talk about the microbiology part mainly. So before this, there was a slide. Oh no. Okay, the slide is being probably deleted or whatever. So there were so many mathematical formulas regarding those determination of the biodiversity that is not so relevant for at this moment. That calculate Shannon index, a kind of index, relative frequencies of different plants, the biomass, the abundance of different plants. So if you see the result out of that, this I am showing only for the Haspahari forest, and that the number of plants and the different plant varieties, the trees that were present in the forest. It is in percentage. It is being shown that we can have a visualization in the pie chart that a particular species is taking how much space. So it is basically sal forest. 12% of the plants are sal plants. And the others are also present. There are a lot of climbers. There are Pial, there are Kasmula, Mohua. You can, in Bengali, there are Arjuna, Asan Gach. They were present relatively in less number compared to the Sal. And there are so many climbers and Uri climbers that we call lioness. So this forest has hundreds of different kinds of plant varieties. And this is the importance value index. I think the next picture will help you to understand. This is the man-made forest, the IVI of man-made forest. It comprises mainly of acacia auriculiformis, akashmoni or sonajuri, that is called. These are the plants growing under the canopy of this akashmoni forest. These are basically herbs and shrubs. They are not trees. The only tree is this akashmoni. And if you see the abundance, which is a better picture about the distribution of the different plants, the sal forest, sal comprises of 75% of the forest area of that pristine natural forest. And the other plants, they comprise the 25%. But the diversity was quite huge. But here, if you see the Akashmani, it comprises almost 99.9% as far as the abundance is concerned. Abundance includes the diameter of the plant also. That's why this increased. As far as when we are seeing the numbers, by numbers, you cannot actually estimate which plant is dominant, which plant is not so dominant. So for knowing dominance, you need to know the abundance. So anyway, so it is quite clear that the natural forests have a very huge biodiversity, species richness in comparison to the man-made forest. So then we did the elemental analysis of the floor, of the forest floor that is being formed year after year, the existing nature of the floor soil. We did an elemental as well as some physicochemical analysis, some enzymatic analysis like acid phosphatase, dehydrogenase assay. And this is called substrate-induced respiration assay. Phosphate, carbon, and nitrogen, the contents were determined. And in all the aspects, this was called EDX, energy dispersive X-ray spectrometry, by which you analyze the elements present in a particular material. So by this analysis, we came to a conclusion that the man-made forest is poor in nature of nutrient cycling, and also in terms of microbial biodiversity. It is poor in nature in terms of plant biodiversity as well as microbial biodiversity. And there are a number of heavy metals also present in the man-made forest. They are more concentrated. They're also present in the natural forest, but in the man-made forest, their concentration is high. So that is harmful. This comparison was done just to understand the nature of the forest. And then next year, we were going to study the successional stages of the leaf litter decomposition. This was the main target of our study, main purpose, that how leaf litter is being converted into soil. Soil is the main thing here. Successional stages here means about the microorganisms as well as the chemistry and the physics of the material that is being changing time to time in a time course manner. So for that, we need to have a leaf litter that is being immobile. That mean, leaf litters are always present in the forest, but you have to have that leaf litter which you have placed there. And if the leaf litter is going to move to other places or mixing with the other leaf litters, you are not going to have the same leaf litter which you have put. For that reason, you need to put the leaf litter in the forest in a trap. And I made a trap of a mosquito net comprising of 12 by 12 inches size, and I put these traps at various places inside the forest, and started its study from zero day onwards to the day when it is converted into the soil. This was the target. And this is the man who actually helps me while going to the forest. This is the different pictures about the forest. Once I went here inside, there is a lake. Also, the small pond, you can say, not a lake actually. So these kind of traps were placed inside the forest, and we picked up such bags of traps every time we visited the forest and brought into the laboratory and did different kind of experiments out of this to understand how the leaf litter is being gradually converted to soil. One of the study included the samples that we could collect from the forest as the leaf litter traps were 7 samples we could collect from 0 day onwards to 280 days. Until then, the 90% of the leaf litter had been, I would not say 100%, 90% had been converted into soil in 280 days. So approximately 1 year it takes to convert in this forest, which is a deciduous forest, and the rainfall is not that very high. The annual rainfall will go about 1,000 millimeter per year. So a little bit drier area, you can say. So one of the things that was the target was to study the microorganisms that are coming in every sample. So there were 2 approaches actually. One of the approach is culture-dependent approach. The another one is culture-independent approach. Here I will talk about the culture-dependent approach. In culture-dependent approach, we used various kinds of media to screen the fungi, the fungal strains, as well as the bacterial strains. The fungal strains were, these are the numbers on the basis of the morphology of the colonies, and that they seemed to be different with our observation. On that basis, we could see that there were more fungal strains in the beginning of the leaf litter, but when it was gradually converted into soil, the number of bacterial bacterial types, or you can say strains or species, increased. So at the initial stage, the bacteria and fungi come into play, and they gradually transfer the job of decomposition to the bacterial strains. But this is not the only observation which can convince our our hypothesis. This was a result, not hypothesis, but people also have found a similar kind of results, that at the initial stage when the leaf is drier, in drier environment actually fungi can grow. When the leaf becomes a little bit wetty and there is a moisture inside, then only it can support bacterial growth. And that was the thing also happening in our study. So we did some physicochemical analysis of those samples from 0 day, 30 days, 60 days, 90 days. Each of these things were gradually changing. I will not go into details of how they were changing, why they were changing. It will be a research like talk. It will become. Just I'm saying that these things can be done to understand the process of leaf litter decomposition. So electrical conductivity, the change in the pH, the moisture content, and mainly the carbon, phosphorus, and nitrogen content. They tell many things about the nature of the conversion or decomposition, how it has been going on. One more thing is this lignin content, because the plant material that contains lignin as the most recalcitrant substance to be degraded. So this is very essential to understand how lignin content is decreasing. So in every 30 days or 60 days interval, the lignin content got degraded. These are three lines over here. They are basically, this is the acid soluble lignin, this is acid insoluble lignin, and this is the total lignin. So lignin content tells about how the leaf litter or the plant material is being degraded gradually. These are some enzymatic activity that tells us about the microbial activities going on in the leaf, in the litter. So we can see that the most microbial activity was found to be during the 60 days, 90 days, these periods. At the initial stage, it was less because the leaves were dry, but in the middle, the microbial activity were quite high. And when it was gradually converted into soil, the activity gradually decreased as far as this dehydrogenase activity is concerned. This is one enzyme, which is the most important enzyme by which we measure microbial activity. And then when we added the antibiotics, antifungal and antibacterial antibiotics, we could see that the role of, actually to understand that this dehydrogenase activity is because of what? Because of bacteria or because of fungi. For that reason, you used to use these antibiotics. And by that, you understand at the beginning, at this stage, at 60 days stage, the bacterial activity was quite high. Now this is fungus. Fungal activity was quite high, quite natural. At the beginning, fungi were more in number, bacteria were less in number, but fungal activity also decreased, as well as the bacterial activity also decreased in the middle and later on, as far as this dehydrogen activity is concerned. Regarding acid phosphatase activity, it gradually increased from 0 day onward to 280 days onward. And the same kind of study, that antibiotic, antibacterial, antifungal antibiotic, and the pattern is quite similar in both the cases, you can say. This enzyme lacase, this is very, very crucial, and about the industrial microbiology, I will talk a little bit about this later on. That lacase enzyme is very crucial in this kind of environment where a particular substance is being, like lignin, is being broken down into smaller products. So lacase activity was measured from 0 day onward, and it was found to be gradually increasing and maximum at the end 150 days, and then suddenly it fell down, and then this was a little bit higher during 280 days interval. And then we measured this is very interesting. This experiment can be done anywhere in the college, and this tells us very important things about the microbial activity that is going on. This is called substrate-induced respiration, an old experiment, substrate-induced respiration. What is it? This is the diagram that illustrates this process. What is done, you take some soil sample or whatever sample of which you want to do. We want to understand the microbial activity present there. One of the process is enzymatic activity measurement, but you can also measure the respiratory level. Respiration is a measurement of metabolism, definitely. 10 grams of soil was taken in a conical flask. This experiment was designed by me. 10 grams of soil mixed with, say, a particular substrate. The substrate could be anything. from simple to complex. Here it was kept as glucose as a substrate. This glucose will help the microorganisms present here to grow a little bit and to do their activities, metabolic activities, to enter into the metabolism. So if microbes are present, they will definitely enter into the metabolism. If they're less in number, the less metabolism will take place. And this test tube contains NaOH solution, and this flask is completely sealed with rubberized plugs. So what happens, the microorganisms present in the sample, they will respire, they will release carbon dioxide. That carbon dioxide will be absorbed by the NaOH present in the tube. This is open. And the NaOH will be converted into sodium carbonate. And the remaining NaOH, which will not be converted, which will remain inside because the concentration is 1 molar and it is quite high, quite a bit in amount. Not the entire NaOH will be converted to Na2CO3. So if it happens, you increase the amount. So remaining NaOH can be titrated by HCl to understand how much NaOH is left inside. So but that tells you about how much NaOH is converted into Na2CO3. So Na2CO3 is directly proportional to the carbon dioxide that is being released by the bacteria. So it tells you indirectly about the metabolic level or the metabolism that is going inside the sample. So this is a beautiful experiment. You can change the substrate and you can do different kind of studies with this. And we also put antibiotic into it to understand whether this metabolism is bacterial or fungal. So when we took the total study without adding antibiotic, it was found to be gradually increasing. The metabolism rate was gradually increasing from 0 day, 30 to 60 day to 280 days onwards. But when we put antibiotic into this fungal antibiotic, the fungal antibiotic to kill fungi and to allow bacteria to respire. So the same was the result. At the initial stage, we saw that the fungi are more contributing to this metabolism, and in the later stages, bacteria are contributing more to this metabolism. So these are a kind of environmental studies, you can say, by which you can understand a particular nature of a soil, of any sample, of agricultural land or any polluted land, whatever you want, you can study like this. This is a study, it is called phospholipid fatty acid analysis. By this, you can understand the nature of the microbes that are growing there. There are certain marker fatty acids by which you can identify which kind of microorganism is predominant at which stage. So this helps you to understand, go into little detail about the successional stages of the microbes that come from 0 day onwards. So this is the tabular data. It will be difficult to interpret from this, so I made it simple like this. From those PLF analysis, what we can conclude, the samples were put into the traps were put into the forest in the month of March, at the beginning of March, and 280 days were completed about in December or so. So during this period, how the leaf is being converted into soil, that if we see the microorganisms involved, with the help of this fatty acid analysis, we find that similarly the fatty acid cells tells us that at the initial stage the fungi were more in number and they gradually declined. Gram-negative bacteria, they picked up later on. Anaerobic bacteria were present all along, with a little less during the initial stage and more during the later stages. Hyphal fungi, there are certain marker fatty acids that indicate that these are hyphal fungi. They came into play during June, July, August, September, this period. And another bacterium called Springobium, that was also found during this June to August or September, during this period, to grow and to convert. I don't know what process these were involved. It's difficult to say what they were doing inside this leaf litter. Since they have come, they must be engaged in decomposition process that can be imagined. So from here, I will talk about the bacterial and fungal cultures that we isolated, what we did with those fungals. From here, we enter into a little bit about the industrial as well as environmental microbiology. So, or agricultural microbiology, you can say. with the bacteria that were screened, we subjected them about hundreds, more than hundreds, in fact, bacterial strains and more than 100 fungal strains were isolated screened from the leaf litter during this couple of years of research. And all the bacteria, I gave there were two fellows. One fellow was given this responsibility of hand to handle bacteria, other fellow was given to the fungi. So at the initial stage, we did the PGPR, plant growth promoting rhizobacterial characteristic. We wanted to see that because this is I had been doing PGPR research quite a long time. So I was interested to get some strains which are potent PGPR. So there are some some traits, PGPR traits, which actually tell you whether your bacteria. Just a minute. Who is this? Just a minute. Sorry, I stopped it. The PGPR traits were analyzed. PGPR traits includes different characteristics like amylase, protease, cellulase activities, phosphate solubilization, ammonia production, IAA production, ACC deaminase activity, siderophore production, and salt tolerance. And we also saw some things like ampicillin and chloramphenical resistance property. We also saw heavy metal properties, heavy metal resistant properties. And we also applied those bacteria to rice plants and in presence and in absence of heavy metals and the bacteria. These are you can, by these experiments, you can understand 0 is the control rice plant. This one is the particular heavy metal without the bacterium. And this is with heavy metal plus the particular bacterium, which was resistant to that. 3 different heavy metals have been used: cadmium, lead, and arsenic. So I have shown just a few pictures of that. There are so many different kinds of similar kind of. You can see that the bacterial inoculum, when it was added in all the cases, in fact, the growth was, in certain cases, it was better than the control, which contains neither heavy metal nor bacteria, 0. So these bacteria are very good plant growth promoting rhizobacteria. And as far as the fungal strains that were screened, they were subjected to study their decompositional abilities. For that also, there were some enzymatic analysis like amylase, protease, cellulase, and the most important one, this lacase activity. And that was also done. Phosphate solubilization was also seen because at the initial stage, I thought to do plant growth promoting fungal. I wanted to screen those, but later I shifted my idea to decomposition to find out fungal strains that can degrade recalcitrant toxic materials. So we subjected the fungi to some dyes present in our lab, like bromophenol blue, Congo red, alizarin red, like this, and we came out with these are the chemical structures of those dyes, like bromophenol blue, Congo red. These dyes are used in textiles, and they are very, very toxic. In textile industry, at a certain stage, the dyes are being bleached. For bleaching, they use various kinds of chemicals. So if you use this lacase produced by this fungi, and those chemicals, we can get rid of those chemicals that are ultimately going into our water bodies and affecting us. So these were the experiments by which you could understand that how these fungi are able to degrade Congo red, bromophenol blue, alizarin red, etc. They were highly potent. So then we tried to study when and how they are producing the lacage. The time course study was done at different pH for these 6 strains only. And we came out with this idea that it is from day 6 onwards, the lacase enzyme is being produced in most of the cases, except a few like this one. It produces 7, 8, 9 days. This one produces at the 10 day, it produces the maximum lacase activity shows. So the optimum pH was found to be 5 in most cases. 3 and 4 were also for these. These are the names of the lab designated strains and that fungi were used. And when they were compared as far as their lacase activity is concerned, this particular strain, H9PF2, was found to have the maximum lacase activity. So we sent them for identification, 18S rRNA-based identification, and from that, 2 strains were found to be contaminated according to the NCMR authorities, but we had sent them the pure culture. We had pure culture. Out of that, 4 strains could be identified, and 3 were found to be similar, Aspergillus costariciensis. The 3 of them were, but I will show that they are a little bit different among each other. Another one was Fusarium putens. So we did a phylogenetic analysis using MEGA7, and then I did an alignment also. In the alignment, I could find that the alignment, this is very small diagram, is very difficult to see. Each of these strains are different. Here you can see this one is different from this one. There is a gap over here. Other parts, it is quite similar. So the name Aspergillus costariciensis for the 3 species, I don't know whether at the species level they could be. The blast shows that they are Aspergillus costariciensis, but there are differences at the level of sequence, 18S rRNA sequence also. These are among 2. This one is again here having different sequence from that one, and it has gaps at different areas. So these 4 strains which were sequenced, the Fusarium is absolutely different, but the other 3, they are also different strains of Aspergillus costariciensis. So that is all about the research associated with this industrial as well as environmental microbiology. Thank you very much for your patient listening. Thank you. Hello.
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 Hello. Can you hear me?
Mm-hmm. Yes.
Okay. Thank
course,
you, sir,
madam.
Thank you, sir, for enlightening us with the soil microbial activity, the environmental biotechnological part, and the industrial microbiological part of this session. And you discussed about the enzymatic activities while decomposition is taking place. You discussed about the fungi as well as the bacteria which took part in this decomposition process. You told us about the marker fatty acids which were present during this decomposition, as well as you told us about certain gram-negative bacteria and about certain sphingobiomes which were responsible for this decomposition process. And you also discussed about the plant growth promoting bacteria, their lactase activity, and about the fungal dyes which were produced. Thank you, sir, for this enlightening speech.
Thank you very much. Your analysis was very nice. Thank you.
Okay, sir. And over to Dr. Rituparna Kundu Choudhury for the questionnaire session. Hello? Dr. Rituparna Kundu Choudhury? Hello? The students can as well ask any questions, any doubts they can hear from the sir. Students, can you hear me? Do you have any questions?
I can see one question asked by Onik Banerjee.
Okay, okay, sir.
Is there any difference between detrivores and decomposers? Yeah, detrivores at certain level they are similar, but detrivores are usually you can say insects and worms. Those kind of organisms are mainly the detrivores. They comprise the detrivores. Small insects also there. This is and decomposers are which convert them into at the mineral level basically. So detrivores are at the initial stage definitely involved, and that part has to be studied along with this by someone. because there are nematodes, there are worms, there are other insects there. At the initial stage, they're also chewing the leaf material and other litter that is being falling, and they're converting them into smaller products. Then the fungal strains. Also, you can say some protozoa might be involved. And we have looked to protozoa to some extent by applying antifungal as well as antibacterial antibiotic. I did not talk about that much. But the insect part, the other detritivores, that needs to be studied, of course. Thank you very much.
Any other questions?
Thank you, sir.
Students?
Am I?
Mhm.
Yeah.
Actually, I had a connection problem, I think so.
Okay.
But sir, for your lucid explanation, I do have a question for you.
Mhm.
That the plant growth promoting factor, that plant growth promoting bacteria you have observed rise that were experimentally observed, were they isolated and
Isolated and?
characterized?
Yeah. Yes, isolated and characterized. And they have been applied to rice models also. I just used some of these rice, some of the photographs. Yeah.
Yes. What are the nature of the bacteria? Was it gram-positive or gram-negative?
Different. There are hundreds of bacteria. We talked about. Here I put only some. Now there are 20 bacteria that we have found to be more potent than the others. But during this process, we did an excavatory screening. Whatever bacteria is coming, you keep it one strain of that, and then we will see it later on. Identification part at the classical level has been done, and there are gram-positive, gram-negative mixture of that. 16S rRNA is still yet to be done. I can tell you. On that basis, I am not going into the identification part, which bacteria. There are still gram-positive as well as gram-negative, both types of bacteria.
Thank you very much, sir, for this your very informative session. I think
Thank you.
to make room for your deliberation. Thank you again very much, sir. Now I would like to hand over the microphone to our head of the department, Dr. Pintu Banerjee, and would request him to present the facts.
Over to Dr. Pintu Banerjee.
I cannot hear him properly.
Hello, am I audible?
Now you are audible.
Okay. It's my great pleasure to express vote of thanks for this state level webinar entitled A Voyage to the Industrial and Environmental Microbiology. So, and it's a great opportunity to convey my sincere gratitude and extend thanks to the speaker, Dr. Sandeep Pandey, the Associate Professor of Banwari Lal Bhalotia College, BB College, Asansol. Actually, Dr. Sandeep Pandey is like my brother and junior to me
of the same college where I have studied. I have also studied from Asansol BB College. So, and Dr. Pandey very nicely discussed the leaf litter breakdown and the formation of soil, and also the bioremediation part, and also the PGPR and all those things which are very important and also very much will be very helpful to the students. So I once again express my thanks to Dr. Pandey.
Thank you
When
very
I
much.
asked him, Dr. Pandey, can you please give a talk before our students? Dr. Pandey told me, yes, of course, why not? And I felt very glad that he accepted our proposal. So thanks, Sanjeev.
Thank you very much, Pinto. You're always welcome.
So now I shall express my gratitude and thanks to our Honorable OIC sir of this webinar, Dr. Shobhan Niyogi sir. With his huge administrative workload, he always supported us to organize such type of academic activities and always encourage us. And I extend my heartiest thanks to Dr. Amorish Mondal, IQAC coordinator, who always inspired us to organize such type of webinar. And also I want to acknowledge our advisory member, Dr. Kalidas Das, for his creative and constructive suggestions. I shall convey my thanks to the coordinator of this webinar, Dr. Sutapa Sannal and Dr. Kaushik Sarkar, at the last of my speech. Now, I want to convey my thanks to the convener, Dr. Surmishra Maiti. Dr. Maiti has organized this webinar in a very critical situation when her father admitted at BM Birla Hospital suffering from a heart attack, and her father pacemaker has implanted, and during that period outside the BM Birla Hospital, Dr. Maiti has contacted different speakers and chalked out the plan and program of this webinar. So for this, I especially thankful to her because I told her that please organize such type of webinar for the sake of the student, so that we can enlighten the student for industrial and environmental microbiology, as they have in their syllabus, in their DSc syllabus. So we will have to organize such webinar from different parts of the country, and as we have some eminent speaker, and Sanjeev Pandey is one of them, just very near to us, and that's why we called him. And another convener, joint convener, Dr. Rituparna Kundu Chaudhary also helped us, also helped to organize this webinar in every respect. So many thanks to her. And also I want to convey my thanks to Dr. Sumanjana Khatua as the organizing member of this webinar. Now I want to express my thanks to the coordinator, Dr. Sutoba Sannel and Dr. Kaushik Sarkar. Dr. Sutoba Sannel, from the very beginning, she is with us to organize such type of webinar with our department, and she helped in all respect in organizing every webinar. And Dr. Kaushik Sarkar, without whose support and without whose technical support and technical help, we cannot organize such type of webinar. We will organize another 3 webinar, another 3 lecture series, and this is the first lecture series for the industrial and environmental microbiology. This is first lecture. Another 3 lecture series we will organize, and all these 3 lecture will be organized by the help of Dr. Kaushik Sarkar. So how can I pay Kaushik for his kind help, or by a simple thanks? So a simple thanks is not enough for Kaushik. And also Dr. Krishnendu Sarkar, who is the technical advisor of this webinar. So without the help of Dr. Kaushik Sarkar and Dr. Krishnendu Sarkar, we could not be organized such type of webinar. Last but not the least, the participants, that is our beloved students who have participated in this webinar. This webinar would not be successful. So I convey my thanks to our beloved students. So, thank you all. Thank you, my brother Sanjeev.
Thank you, Pinto.